Students, which is the energy currency of the cell? In eukaryotic cells, which is the organelle which is called energy currency of the cell? Which one? Options: chloroplast, mitochondria, ribosome. Which one? Yes, ma'am. Tell me, which is the organelle which gives ATP, stores ATP? Tajuddin, I've given the options. Which is the energy currency of the cell? Which organelle? At least I'll give you two choices, answer in that. Chloroplast, mitochondria.
Mitochondria.
Mitochondria. Mitochondria is called energy currency of the cell because mitochondria has electron carriers. Okay. So, if I talk about eukaryotic cell, in the eukaryotic cell, it is the mitochondria responsible for generating ATP. Left side also appears in the mitochondrial matrix. Okay, here in this inner mitochondrial membrane, in the inner mitochondrial membrane, you will find the electron carriers. Okay, in case of prokaryotes, this is eukaryotes. Okay, this is eukaryotes. You can see here. In case of prokaryotes, what is the condition? It is a bacterial cell, an inner cell membrane. Okay, so where is the ETC occurring? In the plasma membrane. In the plasma membrane. Okay, here only ETC occurs. So, in a prokaryote, that is in a bacteria, okay, we can expect the ETC, electron transport chain, occurring in the plasma membrane. In case of eukaryotes, inner mitochondrial membrane. Understood, all of you? Basic things. Okay. Now, what are the things present? What are these? Electron carriers. There are electron carriers. Okay. These electron carriers are very important because they very simple. They carry electrons or protons. Electrons and protons. There again you have differences, no? For example, if I talk about flavoproteins, right? FMN, FAD, these are the flavoproteins. Okay, they can carry both. They can carry both electron and proton. If I talk about quinones, ubiquinone, naphthaquinone. In photosynthetic organisms, in light reaction, you have plastoquinone, but of course not in general ETC. This is in specifically chloroplast. You can find this plastoquinone. They will carry both electron and proton, right? Coming to iron sulfur protein, they carry only electrons. Similarly, cytochromes. They carry only electrons. All of you understanding this basic things? I've written on the board basic things. Only 17 have joined. What happened to remaining? Morning? What about the morning session, Sahana?
So there were people, sir.
They were. Now they are not there.
Yes, sir.
Okay. I'll send you this one second. I'm taking a photo. Okay. So what do you understand here? There are different types of electron carriers. Okay. These electron carriers are responsible. They're mainly responsible for transferring electrons from one electron carrier to another electron carrier by the process of reduction and oxidation, OR reactions. Reduction and oxidation happens. Okay. Now let us understand basically how these electron carriers are situated. Okay. I've just named them now on the board. We will discuss them in the form of like how they are actually present, what is their role, etc. So let us draw the diagram. If those are missing the class, they should draw the diagram and keep it next class. At least let them follow it up. I'll be starting with the electron transport chain now. We will be discussing each electron carrier also a little later as the class goes. Okay, so let us draw the diagram of the cell membrane. See, I will draw the cell membrane diagram in case of eukaryote cell membrane, which is going to be the inner mitochondrial matrix what we will be discussing. In case of bacteria, we will be discussing its related to plasma membrane. Understood? Okay. So, coming to the diagram of ETC. Okay, if I go here and check it out now. So here is the phospholipid layer. Okay, these are the phospholipid layers. The phosphates are drawing first. These are the phosphates. Okay, yeah, these are the phosphates. And let us draw the lipids. These are the lipids. I'm drawing few places the lipids. Just show it here and there, just to represent it. Okay. Now let us write the various electron carriers. So first comes FMN or FAD. FMN or FAD is the first electron carrier. Followed by this is the iron sulfur protein. Next comes COQ or ubiquinol, both are same. You can call it coenzyme Q or ubiquinol. There's another iron sulfur protein, FES. Okay, followed by this is another ubiquinol. Another ubiquinol. UBQ means ubiquinol. So FMN or FAD, iron sulfur protein, ubiquinol, iron sulfur protein, ubiquinol. Okay, then comes cytochrome B, cytochrome C1. cytochrome B, cytochrome C1. Followed by this is cytochrome C. Here it will be above the phospholipid. Cytochrome C. Followed by this is cytochrome A1 and cytochrome A3. Followed by this is cytochrome A1 and cytochrome A3. Okay, so to look into that once again, FMN or FAD, iron sulfur protein, ubiquinone, iron sulfur protein, ubiquinone, cytochrome B, cytochrome C1, cytochrome C, cytochrome A1, cytochrome A3. Okay, let us continue this little more. Show the ATP synthesis channel. After this comes the ATP synthesis channel. Okay, let us not write a small diagram, rough diagram at least we will write. In UG, simply we'll draw one line, one channel. Here, let us show something. Later on, we can go detailed structure of the ATP synthesis channel. Let us write at least like this. Let us not name them everything now, at least write something like this. Okay, at least write like this. This is the ATP synthesis channel. Okay, so here we should understand the concept of electron transport chain. Can you help me out? Just now I was telling you which is electron carrier alone, which is proton electron carrier both. Let us understand that. Just try to guess. Any one of you can unmute and tell. Let us see. FMN, FAD. Both electron carrier plus proton, or it's only electron carrier? Just tell me what it is.
So both, sir, electron and proton carrier.
Okay, both. FES?
It's only electron.
Okay, electron. Ubiquinone?
Both electrons and protons.
Okay. Again, FES comes only electron. Again, ubiquinone, proton and electron. What about these cytochromes B, C, C1, C, A1, A3?
Carry only electrons.
Carry only electrons. Okay, it was a revision for you once again. Anyway, now all of you understand from Krebs cycle and glycolysis we got reduced coenzyme. Which is that one? Which is that reduced coenzyme we obtained during glycolysis and Krebs cycle? Something we got now from glycolysis and Krebs cycle. What was generated from here? What is that? Reduced coenzyme, which one?
NADH2, sir.
Yes, correct. You're right. NADH2. So NADH2 is donated to FMN and it becomes NAD. NADH2 becomes NAD. How do you call it? What do you call it? It oxidized or reduced?
Reduced, sir. Reduced. Reduction.
NADH2 is reduced. Whenever it's receiving hydrogen, it reduced. This got oxidized. NADH2 got oxidized to NAD. NADH2 donated it to FMN. Which one? Hydrogen to FMN. FMN became FMNH2. What is that then? That is called. FMN received hydrogen. There it is reduced. Understand here. FMN reduced to FMNH2. NADH2 oxidized to NAD. I'll repeat once again. NADH2 oxidized to NAD. Okay, in the same process, FMN got reduced to FMNH2. Okay, see the top diagram on the top side and write that how to understand that. NADH2 plus FMN became FMNH2 plus NAD. Did you understand this reaction? NADH2 plus FMN means NADH2 plus FMN gave FMNH2 plus NAD. That means what? Hydrogen from NADH2 was received by FMN to become FMNH2. So NADH2 oxidized to NAD, FMN reduced to FMNH2. Are you clear? Did you understand this reaction? You list out the reactions, okay?
So can you please repeat again, sir?
Once again, I'll tell you, okay. See the board. NADH2, yeah, see the diagram. You're showing the diagram also. NADH2 plus FMN. NADH2 plus FMN became FMNH2 plus NAD. That means hydrogen was donated to FMN to become FMNH2. NAD became free. That's what is shown here. Got it? Basically. Now, that means what? NADH2 oxidized to NAD, FMN got reduced to FMNH2. Whichever receives, it is reducing. Did you get the concept? Yes?
Yes, sir.
Okay. Now, let us show the diagram like that. Here, how do we show this in the diagram? See the board. See, this hydrogen is there, no? That is given to FMN. Correct. So, this became FMNH2. Back it became FMN. Okay. This hydrogen was converted to proton and it is pumped across the membrane. Because hydrogen got to proton to electron, no? Electron was transferred to the phase. Yes, all of you understood here? Okay. Electron was transferred to FES. Now, FES got reduced. FES got reduced by accepting electron. Okay. And it donates electron to COQ. Donates electron to COQ. But then COQ got reduced. By accepting hydrogen, ubiquinone got reduced. Okay. Understand that? Ubiquinone accepted two protons from cytoplasm. See? It accepted two protons. Okay. Then how do you understand this reaction? I'll write it this side just for following up. I'll erase it afterwards. You don't write immediately below the diagram. Write it elsewhere. First reaction, what I wrote on the board. Second reaction, I'm writing there. Observe. Ubiquinone is just accepting two electrons and two protons, and it becomes UbQH2. UbQH2, it's forming. Okay, this is reduction. It got reduced. Again, what will happen? UbQH2 gets oxidized. Oxidation. How? Converting back to ubiquinone plus 2 electron plus 2 proton. Okay, understand here. These 2 protons are getting pumped across the membrane. Pumped out across the membrane. And 2 electrons are transferred where? To continue the ETC. To continue ETC, they will go ahead. Next means FES comes next. Okay, all of you are clear in the reaction? Till here, understood? Yes?
Yes, sir.
Okay.
Sir.
Yeah. Any doubt in this, Manasa? Okay.
No, sir.
This electron is now, that means UP, I'm not showing it again here, I just once we show it here. Same thing here. So just represent two protons coming out, pumping across the membrane. An electron is transferred to FES. Okay, this FES again transfers electron to next electron carrier, which is again ubiquinone. Ubiquinone has the same property, accepting protons. Okay, it becomes UbQH2 similarly. So let us only represent that there is proton pumping happening. Okay, and above this is another outer membrane. If it is mitochondria, this will be outer membrane. Okay, above, top, I just compare. See, if it is mitochondria, this is outer membrane, this is inner membrane. If it's a bacteria, this is a cell wall and this is a cell membrane. Okay, so here is the protons accumulating. From there, it is very simple. What's going to happen? Again, electron is received by cytochrome B. Reduction, oxidation. That's why we'll call it as reduction, oxidation. Alternatively, in many of the information you would have read it. It's called OR reaction, oxidation reduction reactions. Okay, yeah. So alternatively, it keeps happening. So again, this will get reduced by accepting the electron. Again, from cytochrome C1, it reaches cytochrome C. Electrons are transferred. Every time, reduction, oxidation. Again, this is reducing by accepting electron. And ultimately, cytochrome E3 receives the electron. Okay, so this cytochrome E3 will donate electron to oxygen. Understood? That is why we'll call oxygen as the terminal electron acceptor. Terminal electron acceptor means they receive the electron finally. So this oxygen, okay, along with 2 electrons and 2 protons. Correct? So 2 protons from cytoplasm and 2 electrons from cytochrome A3. You can show it like this. Okay, and that is going to help in formation of water. That is why we will say that water is the end product of ETC, electron transport chain. Okay, so till here we have understood that water is going to be the end product of the ETC chain, electron transport chain. These protons which are present here, these protons are going to ultimately create PMF, proton motive force. Okay, and that is going to influence the ATP synthetase channel. What is this called? What is this called? ATP synthetase channel. Okay. This is called ATP synthetase channel, wherein the protons will enter this channel and they influence the various proteins present in the ATP synthetase channel, which is A1 protein, beta 2 protein, then you have alpha, epsilon, delta, alpha, beta sheets. Okay, those proteins will be present that we will discuss in the next session. We'll be talking about the ATP synthesis channel separately. Okay, they're called oxysomes also, other name. Okay, so here as protons influence this proteins that are associated together and ultimately converts ADP, an inorganic phosphate, into ATP. adenosine triphosphate ATP. So here this ATP is forming. Here is the enzyme called ATP synthesis. Here it is present in the base of this. So ATP synthesis channel helps in forming ATP. This much we should understand as a basic when you talk about the electron transport chain in physics. So we will be learning in further sessions, we'll be learning about every electron carrier and their role, okay, the structure and how they will help in forming the ultimately the proton pumping. And the next topic is related to oxidative phosphorylation. Oxidative phosphorylation means how ATP is formed after electron transport chain. After ETC chain, what is going to be the consequences? How the ATP will be formed in this, how the ATP synthesis channel will function in generating converting ADP and inorganic phosphate into ATP. That's what another topic. That's why both are connected. Electron transport chain and oxidative phosphorylation both are connected to one another. Okay. So now all of you, I think hopefully you have followed till here.
Yes, sir.
Yes, sir.
So I need to add on a few more things to tell you. Complex 1, complex 2, complex 3. I'll name them now. I'll just name them. We have some time. Let us discuss that. I'll do one thing. I'll label them down here. I'll label them down one by one. Then we can label it in the diagram. Complex 1. Complex 1 is called NADCO2 reductase. NADCO2 reductase means it starts from NAD. We have the first one now here, NAD. Okay, this NADH to this part. NADCO2 reductase means it starts from here and up to this. Okay, COQ. The complex is called complex 1. Means this particular transfer of the electrons and protons is called complex 1. Followed by complex 2. Complex 2 is called as succinate COQ reductase. Succinate COQ reductase. Okay, this is what is it called? Complex 2. Writing it in brackets. This is called complex 2. So complex 1 is NADCO2 reductase. Complex 2 is succinate COQ reductase. Succinate COQ reductase means it says this proton is coming from succinic acid. So succinate, that's why we call it as succinate. So succinic acid helps in giving these protons. Okay. Succinate COQ reductase means it's starting from here and goes up to COQ. Okay, that is why it's called second complex here. This is called second complex, complex 2. Then complex 3. Okay, complex 3. Complex 3, name of this is called as COQH2 cytochrome C reductase. COQH2 means from formation of COQH2 means ubiquitin here. Okay. 2 cytochrome C. Cytochrome C is here. Here, sorry. From here to this cytochrome C. Okay. That is called the complex 3. So I'll just show that once again. Here. This is the name of the section. Then comes 2 more complexes. Complex 4. Complex 4. I'm writing it here. Complex 4 includes cytochrome C oxidase. In the sense, this will carry from cytochrome C to water. Means this particular thing is complex 4. Okay, so what do you call this as? Cytochrome C oxidase. The role of different enzymes. Cytochrome C oxidase. And finally, complex 5 includes the ATP synthetase. Okay, so complex 1, complex 2, complex 3, complex 4, complex 5. So to show you in the diagram, NAD to COQ, NAD-COQ reductase, complex 1, succinate to COQ, complex 2, succinate COQ reductase, complex 3, COQH2 means from here up to cytochrome C, COQH2, cytochrome C reductase, complex 4 from cytochrome C oxidase it's called, where they help in using oxygen and ultimately giving water. Complex 5 includes ATP synthetase channel. Okay, so we have learned some basics on electron transport chain, the basics of the classes like this, where you have understood that the various electron carriers present and as ATP synthesis channel, they'll all help in bringing the ATP formation, which includes two things. Number one, what is it? Electron transport chain, followed by oxidative phosphorylation. All of you understood this topic? Basic?
Yes, sir.
Okay.
Yes, sir.
Okay. So what we will do is, okay. So students, now what we will do is, we have to learn each electron carrier. We have to learn each electron carrier in the sense structure of FMN, FAD, that one, followed by FES structure. Then quinones. In quinones, we have to learn basic quinone structure because your answer will have weightage. Okay, that's more important. Then we'll be studying about cytochrome structure. These all help you in understanding overall about the electron transport chain. So your answer will be having weightage when you answer all these with the structures. Okay, and finally we will be studying about the ATP synthesis channel also, that is oxidative phosphorylation part. So here in the ATP synthesis channel, we divide this into two parts, F0 fragment and F1 fragment. So also another information we will be learning about the F0 and F1 fragment. This one, again to tell you one more point on this, F0 fragment and F1 fragment, in case of prokaryotes, it's embedded inside the membrane. It's embedded inside the membrane. Whereas in eukaryotes, F0 is inside the membrane, F1 fragment will be suspended in the lumen space where ATP is found. That is why if you see the diagram of the ATP of the mitochondria, this is a crystal. Okay, so here will be the ATP synthesis channel like this. So this is the membrane. You can show like this. This is the upper part and lower part. So here is the ATP synthesis channel inside. Okay, F0, F1, like that we should follow. Okay, so we will be learning about that in coming classes. We will learn this. Next class we will study about electron carriers first, then we will go to oxidative phosphorylation part. So we have 1 or 2 minutes more. Sahana, take the attendance mark. You want the
Yes.
photo of this? Have you drawn this picture? Shall I send you?